Genomes and understanding DNA. The human genome, typical of the genomes of all multicellular animals. It consists of two parts: first nuclear genome, second mitochondrial genome. First, nuclear genome contains 3,235,000,000 base pairs of DNAs. It is divided into 24 linear molecules, each contained in a different chromosome. 48 million shortest base pair, 250 million longest base pair, 22 autosomes, and 2 sex chromosomes X and Y. 45,500 genes are present in human nuclear genome. Second, mitochondrial genome. A circular genome contained 16,569 base pairs, 10 copies present in a mitochondrion. It contains 37 genes. Red blood cells lack nucleus, so they don't have human genome. All our body cells, except gametes, have 46 chromosomes. These cells are called somatic cells. Gametes or sex cells carries 23 chromosomes, 22 autosomes, and one of the sex chromosomes, either X or Y. Each cell carries 2,000 to 7,000 copies of mitochondrial genome. Each female oocyte carries 1,000,000 copies of mitochondrial genome. This is a figure showing a human cell with, in the center, nuclear genome and a mitochondria showing mitochondrial genome. Genome expression. A genome expresses itself first in the form of a transcriptome, then into a proteome. A transcriptome is an RNA molecule formed by the process of transcription reaction. Then a proteome is generated by the process of translation reaction, in which the RNA molecule copies itself into a protein. First, genome. By the process of transcription, transcriptome is formed. RNA copies of the active protein coding genes. Then by translation, proteome is formed. Proteome is the cell's repertoire of proteins. DNA. DNA was discovered by Friedrich Mischer in 1869. He prepared a pure sample of nucleic acid from salmon sperm and showed that DNA is acidic and rich in phosphorus. Oswald Avery, Colin MacLeod, and Maclean McCarthy showed that DNA is the transforming material. They took harmful bacterial cell extract of Pseudomonas pneumoniae and mixed with harmless strain of Pseudomonas pneumoniae. Due to this, these harmless bacterial cells turned in harmful pneumonia-causing bacteria. This proved that bacterial genes made of DNA. Alfred Hershey and Martha Chase used radio labeling to show that when a bacterial culture is infected with bacteriophages, DNA is the major component that enters the bacterial cell of bacteriophages. These above experiments influenced Watson and Crick to work on DNA. These are two figures: A. The transforming principle, B. The Hershey-Chase experiment. DNA is a polymer of nucleotides. First, we should examine what Watson and Crick knew before they began their work. DNA is a linear and branched polymer. It has 4 chemically distinct nucleotides that can be linked together in chains of hundreds, thousands, or even millions of units in length. Each nucleotide in a DNA polymer is made up of 3 following components. First, 2'-deoxyribose, which is a pentose sugar composed of 5 carbon atoms. Second, a nitrogenous base, one of cis cysteine or thymine or adenine or guanine. The base is attached to 1' carbon of the sugar by beta and glycosidic bond attached to nitrogen number 1 of pyrimidine number 9 of the purine. A phosphate group. It is a 1, 2 or 3 linked phosphate units attached to 5' carbon of the sugar. The phosphates are designated alpha, beta and gamma, with alpha phosphate being the one directly attached to the sugar. Structure of a nucleotide. A. General structure of a deoxyribonucleotide, which is the type of nucleotide found in DNA. The four bases that occur in deoxyribonucleotides. A nucleoside equals to sugar plus base. A nucleotide equals to sugar plus base plus phosphate group. Nucleoside triphosphate acts as substrate for DNA synthesis. The full chemical names of 4 nucleotides are: first, 2'-deoxyadenosine 5' triphosphate; second, 2'-deoxycytidine 5' triphosphate; third, 2'-deoxyguanosine 5' triphosphate; and fourth, 2'-deoxythymidine 5' triphosphate. In a polynucleotide, individual nucleotides are linked together by phosphodiester bonds between their 5' and 3' carbons. DNA polymerization reaction involves removal of the two outer phosphates, the beta and gamma phosphate, from one nucleotide and replacement of OH attached to 3' carbon of the second nucleotide. One end of the DNA polynucleotide have an unreacted triphosphate group attached to 5' carbon. The other end have an unreacted OH attached to 3' carbon. The polynucleotide has either 5' to 3' or 3' to 5' direction. The chemical reactions are different in both the direction if it needs to polymerize DNA. DNA polymerization is carried out by DNA polymerase enzyme. This DNA polymerase enzyme is only able to carry out 5' to 3' synthesis. In this direction, only DNA is replicated. Rosalind Franklin, with the help of X-ray diffraction, provided the bulk of the experimental data in support of the double helix model of DNA. Discovery of double helix model of DNA by Watson and Crick, Saturday, March 7, 1953. A short DNA polynucleotide showing the structure of phosphodiester bond. Note that the two ends of polynucleotide are chemically distinct.